All right, let's get right into it. We're about to pull back the curtain on one of the most incredible biological heists you've ever heard of. I mean, this is a crime that's been going on inside our bodies for thousands of years. The mastermind behind it all? The bacterium that causes tuberculosis. And its target? Well, it's our own immune system. And believe me, this is no small-time job. This number you're looking at, 1.5 million. That's how many people die from TB every single year. It's one of the deadliest infectious diseases on the entire planet, and it's stayed that way for over 100 years, despite all our research. So, what's its secret? How is it so successful? Simple. It's an absolute master of deception. You know, the real genius here is that it's like a kind of biological judo. Instead of going head-to-head with our immune system, which is a fight it would probably lose, it tricks our body's defenders. It turns them into its own accomplices. It literally turns our cells into the perfect hideouts. So the big question is, how on earth does it pull off a betrayal like that? Okay, so to really get this heist, we've got to meet the key players. You've got the criminal mastermind, the hijacker, and the high-security fortress it's trying to break into. So on one side, you have Mycobacterium tuberculosis, we'll just call it TB. This is our hijacker, and on the other side, the macrophage. Now, the macrophage is supposed to be this impenetrable fortress. It's a frontline immune cell, and its entire job is to hunt down and obliterate invaders just like this bacterium. The macrophage is supposed to be the hero here, but yeah, the TB bacterium has very different plans. And this is where the whole story gets really interesting, where the mystery starts. Because scientists started noticing something deeply weird. It looked like one of the macrophage's own security systems was actually helping the enemy. Bizarre, right? So this leads to a really mind-bending question, the one that kicked off all this research. How can removing part of your immune system actually make you stronger against an infection? I mean, think about it. Immune receptors are how we spot danger. Getting rid of one should make us weaker, not stronger. But with TB, it was the complete opposite. It was this crazy paradox, and it just had to have an answer. And here's the proof, the actual hard data from the lab. Okay, let's look at the survival graph here. The line with the white circles, those are mice that are missing a specific immune receptor called dectin-1. Just look at how much longer they survive compared to the normal mice shown in black. And then if you look at the other graph, it tells the same story. The mice without dectin-1 had way fewer bacteria in their lungs. This was it. This was the smoking gun. So let's just let that sink in. The crucial takeaway here is that having this immune part, dectin-1, was actually bad for you. It was helping the bacteria. This means our hijacker, the TB bacterium, wasn't just sneaking past the security system. No, it was actively using the security system for its own purposes. The big question was, how? Okay, so the researchers had a new mission. They figured the bacterium must have some kind of special tool, right? A molecular key that it was using to pick the lock on this dectin-1 receptor. Their job was to find that key. All right, so what exactly is dectin-1? The best way to think of it is as a security guard. It's patrolling the outside of our macrophage fortress. Its whole job is to spot suspicious characters, usually fungi, and when it does, it sounds the alarm and triggers this massive immune attack. It is absolutely supposed to be one of the good guys. But here's where it got tricky. This security guard, dectin-1, is really well known for one thing. It recognizes a type of sugar called beta-glucan. You find that on fungi. But the TB bacterium doesn't have any beta-glucan on it. None. So what was going on? The bacterium had to be using some kind of non-standard key, something this guard wasn't trained to look for. And then they found it. The master key. It's another sugar molecule called alpha-glucan. And it turns out the TB bacterium just coats its entire surface with this stuff. It's basically a perfect disguise, like a forged ID badge that it flashes at the dectin-1 security guard. I mean, this quote from the title of the actual scientific paper says it all perfectly. Hijack. This isn't some random accident or a system malfunction. No, this is a deliberate, calculated hijacking. The bacterium has evolved this very specific key for this very specific lock, all so it can live and thrive inside the very cells meant to destroy it. But listen, just picking the lock and getting inside, that's only phase 1 of the heist. Once the hijacker is inside the fortress, its next move is, well, it's even more brilliant. It's time to shut down all the internal alarms. So here's the play-by-play of this sabotage. Step 1, infiltration. The bacteria's alpha-glucan key clicks into the dectin-1 lock. Step 2 is signal manipulation. This triggers a cascade of signals inside the cell that flips a master switch called mTOR. Now for step 3, system shutdown. Flipping that mTOR switch, it completely blocks the cell's internal cleaning process, something called autophagy. And finally, step 4, safe house. With the defenses down, the bacteria can just hang out, multiply, and be perfectly safe. So what is this autophagy thing? Honestly, the best way to think of it is as the cell's self-cleaning crew. It's like the sanitation and recycling department all rolled into one. Its job is to go around, find trash, find broken parts, and most importantly, find and destroy invaders like our TB bacteria. So by tricking dectin-1, the bacterium basically fires the entire cleaning crew, making sure it never gets swept up and thrown out. So at this point, we've got the whole theory of the crime laid out, right? The hijacker uses a sugar key, tricks a security guard, and that guard then deactivates the internal cleaning crew. It's a solid theory, but the researchers, they did one better. They caught the whole thing on camera. They got the visual proof. And the result of all this sabotage is it's the ultimate betrayal. The macrophage, the cell that's engineered to be a bacterium's absolute worst nightmare, gets completely flipped. It's turned into a safe house, a cozy little home where the TB bacteria can live, eat, multiply, all while hiding in plain sight from the rest of the immune system. Okay, and here it is, the visual proof. These are real microscope images. This is what's actually happening. The little green specks, those are the TB bacteria. Now, look at the image on the left. That's a normal cell. Dectin-1 is present and accounted for. You can see the bacteria are just thriving. They're having a party in there. But now, look at the image on the right. This is a cell where they've removed dectin-1. See all that bright magenta? That is the cleaning crew, autophagy, going to town. It's swarming and completely destroying the bacteria. It's an absolutely crystal clear picture of the heist and what happens when you stop it. All right, so let's just do a super quick recap of the perfect crime. The TB bacterium puts on its sugar disguise, that's the alpha-glucan. It uses that to trick the immune system's security guard, dectin-1. That disables the cell's cleaning crew, autophagy, and boom, the macrophage, the killer cell, has been successfully hijacked. So now that we know the hijacker's entire playbook, what does this all mean? Well, it's more than just a cool story about molecular espionage. By exposing the bacterium's strategy, step by step, this research opens up a whole new world of possibilities. I mean, think about it. If we know the key and we know the lock, maybe, just maybe, we can design something to jam that lock, a way to rearm our immune cells and finally, get the upper hand in the fight against one of humanity's oldest and deadliest enemies.